He has served as a faculty for many international conferences like the 15th International Homeopathy Conference at Sri Lanka, Dubai International Homeopathic Conference, London Homeopathic International Conference 2010, and now he is here with us. He is an expert panel member at the Welcome Cure, the largest homeopathic portal in the world. He has worked as a faculty and trainer for the Department of Homeopathy, Government of Kerala, India. Please welcome Dr. Nandakumar.
Thank you.
Hello, says hello.
We welcome the Armenia group. You're waiting for it this morning. These are the papers.
Good afternoon, ladies and gentlemen, and it's a great privilege for me to be here today, and I thank the most respected and my beloved Sharma sir for giving an opportunity for me today. I think you are going to address something totally different which has been said today, something different from what you have heard. There is a paradigm shift in homeopathic practice and research all over the world, and it's a fact. The Central Council of Homeopathy has done so many researches on that aspect. Dr. Kaur has already mentioned about the protocol study of homeopathic drugs. The word specific may be a taboo to many homeopaths. But it has been always been mentioned in different literatures, even from the Hahnemannine era. You cannot forget that. And even today, the patriarch of homeopathy, Dr. George Vidalkas, if you read his famous book, The Essence of Materia Medica, you take the chapter Magnesium muriaticum, he has specified that it is a specific drug for liver disease. So, such type of studies has been done in different parts of the world, and it's more concentrated in the United States of America, where studies has been done with the caladium in prostatic hypertrophy, Crotalus horridus for prostatic cancers, and so on. Homeopathy believes in the energetic plane of existence, I agree. But you cannot deny the fact that a medicinal drug has got a pathophysiological action on the tissues and cells of the body when it is studied using the state-of-the-art technology and methodology, accepting the scientific latest methodological sequences. The time is so much abbreviated and I don't want to waste my time. I'll just jump into what I would like to present before you today. It's a study of the drug Ruta graveolens. The potency is 6 and its effects on brain tumors. Many of you must be knowing about this, I know. But few of you must be knowing the details of this study. The microbiological details, the magnetic study, the electromagnetic study of the cells, the electron microscopic detailed study, and I am going to present you before you all these details today. The study is the Ruta 6 selectively induces cell death in brain cancer, but proliferation in normal lymphocytes, a novel treatment for human brain cancer in the International Journal of Oncology in October 2003. See, I think this is the first of that kind which was published in the International Journal of Oncology and regarding a homeopathic medicine, about a homeopathic medicine. And in this you can see that selectively induces cell death in brain cancer but proliferation in normal lymphocytes. In any study, what we usually take is the metaphase of a cell. In cancer study especially, we take the metaphase of a cell, metaphase of the DNA. This is the normal study which is being done even in the allopathic practice, in the modern medicine, when they research about a chemotherapeutic agent. There will be three things. One is a normal lymphocyte, that's a normal cell. Then a treated cancer, an untreated cancer cell, then a treated cancer cell. So here also I will be presenting before you the three cells. One is the normal cell, one is the untreated cancer cell, and third is the treated cancer cell. Along with the professors of cell biology and the genetics at the University of Texas, MD Anderson Cancer Center, Houston, a paper has been published entitled "The Ruta-6 Selectively Induces the Cell Death in Brain Cancer Cells, Blood Proliferation in Normal Peripheral Blood Lymphocytes, a Novel Treatment for the Human Brain Cancer," and it was published in the International Journal of Oncology in 2003. The method of treating with Ruta-6 and Calcarea foods was followed with excellent results. See, the difference in using a chemotherapeutic agent and Ruta-6 in a brain cancer is that the chemotherapy agent cannot distinguish between a normal cell and a cancer cell. It will just destroy. That's why a patient undergoing a chemotherapy faces a lot of side effects. The amazing part of homeopathic medicine is that this medicine identifies and distinguishes the normal cell, a cancer cell, from a normal cell and produces a damaging effect only on the cancer cell. I will show you the slides. This is the drug Rutagraveolin, you all know, and we have been using this for the past 250 years. This is a graphical presentation of the normal metaphase spread from a control culture. This is a graphical one. What's a metaphase? Metaphase is a coiled nature. It's a coiled type of the DNA where the DNA will be very much condensed. And for all cancer cell studies, for all microbiological studies in cancer, the metaphase of the DNA is being used. So, in this metaphase, there are two things which you have to consider. One, the metaphase the DNA has something called the telomeres. The telomeres are seen at the end of every DNA. It gives a character of the cell. So, if you can destroy the metaphase or you can destroy the telomere, you can destroy the cell, because that particular thing, it establishes the nature of the cell. So, the cancer nature of the cell is being written there. The telomere is seen on the edges of the DNA, which will help the DNA not to stick on to other DNAs, as well as it will prevent the deterioration. So, if you want to destroy a DNA, what you should do? You should destroy the telomere. Let us see what happens. This is the endo-reduplicated and partial metaphase spread showing the dicentric chromatid breaks and tridimer configuration. The time is very less, that's why I don't want to explain more about the chromatid breaks and dicentric. Dicentric means having two centromeres. Centromeres are structures in that DNA. The time is very less, let us go to the next slide. Endo-duplicated metaphase with extensive chromosome fragmentation of the Ruta treated culture. See, this slide. This is a brain tumor metaphase, untreated with Ruta. This is treated with Ruta. You can see the fragmentation of the metaphase. These are graphicals. Now, the most important thing, most amazing thing which I would like to show before you is the original electron microscope pictures. How is it made? These slides show FIS, that's fluorescent in situ hybridization. That is the technology used in all over the world, in US especially for all cancer studies. FIS, and it's the preparation of interface cells from a human B lymphoid cell line and the brain cancer, either untreated or treated with Ruta-6 and Calcarea 4, are stained with DAPI for DNA, that is blue in color, and telomeric DNA with rhodamine. So in the next slide, you just listen to me. In the next slide, you will be seeing a blue structure. That is the DNA. It is stained with DAPI, and in that you can see small red spots. That is the red spots are the telomeres. As I said, the red spots are the telomeres. The blue one, the DNA stained with DAPI, and the red one, it is the telomeres stained with rhodamine. Now see, it is an untreated. It's a control one, is untreated. It is not treated with Ruta. Now just listen to me and just look at the slides. This is the treated DNA with Ruta-6. All the telomeric signs are destroyed. That means the cell is destroyed. The malignant cell is destroyed. Without the telomeric signals, the DNA cannot sustain. It cannot continue its life. So that's how the Ruta-6 acts on the brain cells and produces cell death. And the Ruta-treated human brain cells show significant difference in telomeric signals. You have seen, and the large nuclei from the Ruta-treated cells show reduced telomeric signals. All microphotographs were taken at the same magnification both in vivo and vitro, and the results showed induction of survival signal pathways in normal lymphocytes and induction of death signaling pathways in brain cancer cells. And the cancer cell death has initiated by telomeric erosion and completed through mitotic catastrophic events. These experiments propose that the Ruta in combination with Calcarea 4 could be used for effective treatment of brain cancers, particularly glioma. I don't know, many of you must not be remembering about this. The Times of India has published a Sunday supplement regarding this study, and this paper was the first one, I think the first and the last ever, a homeopathic presentation done in the World Homeopathic Oncology Congress. And I have the paper with me, and that paper, it's written "Sugar Pills Cure Cancer." That was the heading of that Sunday supplement. The Times of India published that. And from these observations, it's evident that these medicines have the definite power to reduce and cure intracranial space-occupying lesions. It may be interesting to note that these claims with regards to the action of these medicines have been successfully vindicated in vitro. These slides show metaphase from control and Ruta-6 treated human brain cells showing mitotic catastrophe. The total number of cases considered for this particular study was 17,324 cases. 17,324 cases. In that, the complete regression was there in 19 cases and improved and static was 21. So just put together, it is 41%. So what homeopathy can offer to brain cancer patients is 41%, which is a huge percentage. Thus, presently, it appears that all these findings, that this new method of treatment with homeopathic medicines may be regarded as the future drug for cancer research and treatment for the benefit of mankind. This is what the American Institute of Oncology says about this study. Let us see how it works. You should know. See, as I told, we have our own philosophy, but it is something written nearly 250 years back. So we should, I personally feel that we should work in this direction also, because we should give a scientific explanation. Scientific in the sense what the scientific world demands from us. If we could give such answers to them, the skeptic criticism can be avoided to a greater extent. And Samuel Hahnemann has given an explanation in par with what was there 300 years back. It's a great science. It's a great discovery. I agree. I always ask when I take classes, I used to ask. Me and my wife went to NASA and saw the aeroplane invented by the
Wright brothers.
It's there. It's kept there. Even today, if you ask any small child who invented aeroplane, what will be the answer? Wright brothers. And next question, will you travel in that plane now? Or are you traveling in that plane now? No, now we are flying in a Airbus A380. But still today, if anybody asks who invented aeroplane, it's Wright brothers. I will, when I finish, I would like to tell you one more line, that later I will tell you. The rooting, the active ingredient of ruta is rutin, and its antioxidant and inflammatory activities, and also for reducing the oxidative damage in a rodent model. And as an inflammatory response, the upregulation of the nitric oxide synthesis INS and COX2, the enzyme, leads to the production of pro-inflammatory mediators, namely the nitric oxide and prostaglandins respectively. The action of the prostaglandin, that's why when we give ruta for anti-inflammatory disease and joint pains, it acts on the prostaglandins. That's why we get relief of the pain reliefs because of the application of ruta. Hence, the significant inhibitory effects on both of these inflammatory medicines unreveal a novel anti-inflammatory action of this plant. In addition, the ruta is also known to protect from the DNA strand breaks and to prevent mutagenesis. That we have already seen. We have seen the slides how the mutagenesis takes place, how the DNA strand is being broken and destroyed by ruta. Let us see how the calcium phosphate acts. It contains phospholipase which cleaves the phosphalidylinositol biphosphate, a membrane-bound molecule that activates protein kinase C. Now you should know what is protein kinase C. The importance of protein kinase C in the management of malignancy. Thus, one may consider these aspects as an explanation to such effects. The protein kinase C has a crucial role in signal transduction for a variety of biological active substances which activate the cellular function and proliferation. The enzyme serves as a receptor for the tumor promoters. Now see, it's a very clear picture of how it acts. Now you can just see the entire action of this is being demonstrated here. The flavone acts on the reaction with the purine and pyrimidine, acts on the DNA. The calcium with calmodulin and calcineurin again acts on the DNA. The phosphate, the phospholipase with AP1. AP1 is a protein complex, again acts on the DNA, and what happens? Spontaneous regression or a prolonged arrest of the tumor cells happens by promoting what? Tumor necrosis factor and resulting to the destruction of cells. That is what is happening in this study and how it works together to destroy the cancer cells. Now I would like to show you two cases, and I have with this protocol I have treated nearly 11 cases, and out of that 4 cases there is complete regression, 3 cases improved and 4 expired or aggravated. Now this is one case. In case number 1, name is Shamsuddin. You can see the video now.
എൻ്റെ ഫാദറാണ് ഷംസുദ്ദീൻ എന്ന പേര്. ഇതുപോലെ ബ്രെയിനിലും ലെൻസിനും ട്യൂമറാണെന്ന് പറഞ്ഞിട്ട് കോഴിക്കോട് കൊണ്ടുവരുന്ന അവിടുന്ന് തിരിച്ചു വിട്ടതാണ്. അതിനുശേഷം ഫ്രണ്ട്സൊക്കെ പറഞ്ഞിട്ടാണ് ഇവിടെ വന്നത്. ഇപ്പോൾ 1 വർഷത്തോളമാണ്. ഇപ്പോൾ ലാസ്റ്റ് MRI സ്കാൻ എടുത്തപ്പോൾ പരിപൂർണ്ണമായിട്ടും അത് ഭേദമായി മനസ്സിലായി. വളരെ സന്തോഷമുണ്ട്.
You can see the previous scan here. It shows multiple ring enhancing lesions. In a magnetic resonance imaging, the cancer cells in the brain is usually written as ring enhancing lesions. As we say that increased echogenicity in ultrasound refers to fatty changes or lesser circulatory stasis there. The same way in MRI it is ring enhancing lesions in the brain. Now this is the previous one before the treatment. Now you can see after treatment. No ring enhancing lesions detected. Again the inference here, you can see no ring enhancing lesions. You can see other few features, that's because of the patient in between had a small TIA, that is transitional ischemic attack, and there was a bleeding from the tumor site. So it was also managed in between. You saw the patient. The second case, and it was the same protocol I continued, because once the patient came to me, he was unconscious, he was just like, no case taking, nothing can be done. Patient has severe aphasia, cannot recognize anybody. So it's no point using the protocol which is worldwide renowned. So I just want to check. So that was used in this case. So the protocol, I will tell you the protocol also. The protocol was Ruta 6, 2 drops BD and Calcarea 4 6 3 green steroids. That is the protocol what is used for this study. Now the second case is pituitary macroadenoma.
എൻ്റെ പേര് അബ്ദുൽ ഹക്ക്. ഞാൻ മണ്ണാർക്കാടുകാരനാണ്. എനിക്ക് സാധാരണ നിലയിൽ ചുറ്റും തലവേദന ഉണ്ടാവാറുണ്ട്. ഒരു ദിവസം തലവേദന അനുഭവപ്പെട്ടു പല ചികിത്സകൾ നടത്തിയപ്പോഴും അതിന് ആശ്വാസമില്ലാതെ നമുക്ക് സ്കാനിങ് എടുക്കാൻ വേണ്ടി ആവശ്യപ്പെട്ടു. സ്കാനിങ് എടുത്തപ്പോൾ തലയിൽ ട്യൂമർ ഉണ്ടെന്ന് പറഞ്ഞു. ഒരു MRI സ്കാൻ ട്യൂമർ ഉണ്ടെന്ന് പറഞ്ഞപ്പോൾ അവിടുത്തെ സർജൻ, ന്യൂറോളജി സർജൻ ഓപ്പറേഷനാണ് ഉദ്ദേശിച്ചത്. എനിക്കത് ഭയമായിരുന്നു, ഞാനതിന് വഴങ്ങിയില്ല. അങ്ങനെ നന്ദകുമാർ സാറിൻ്റെ അടുത്ത് ഹോമിയോ ചികിത്സയുമായി സമീപിച്ചു. അത് കൂടാതെ കുറച്ച് 2-3 മാസം കൊണ്ട് തന്നെ എനിക്ക് ആശ്വാസം കാണാൻ തുടങ്ങി. ഇപ്പോൾ 2 കൊല്ലമായി. 2011 ആരംഭത്തിൽ തുടങ്ങിയ ചികിത്സ ഇപ്പോൾ തുടരുന്നുണ്ട്. എങ്കിലും 3-4 മാസം കഴിഞ്ഞപ്പോഴേ ആശ്വാസം കാണാൻ തുടങ്ങി. ഇപ്പം നോർമലാണ്, കുഴപ്പം അനുഭവപ്പെടാറില്ല.
Now you can see non-enhancing supracellular mass which measures 1.2 into 0.7 separate pituitary gland, pituitary macroadenoma. This is the one previous the scan. Now see this one. Essentially a normal study. In view of the resolution of the supracellular lesion seen on the previous MRI studies, most likely to be neurosarcoid result. The radiologist was totally astonished. He took my number from the patient and he rang me up and said, "Oh, sir, what did you do?" I gave homeopathic medicine. Which medicine? Will you understand if I say that I gave this medicine? No, this is first of this kind. It's first of my experience, that totally resolution. Because I have both the MRI films with me, the previous one and the current one. So, it's totally resolved. Same protocol used. My dear friends, I would like to abbreviate just saying one line. The famous Indian philosopher, Mr. Aurobindo, many must have heard about him. He has said, the last word of nothing in this world have been ever said. The last word of nothing in this world have been ever said. So let us take the last word of Organon of Medicine 6th edition and work together in a different direction and uplift the pride of our great.
Hello. It was 2010 when I met you from our college, and actually it was not scheduled your lecture here. So we had one Dr. John Bennett from America and Dr. Joasha from Mumbai, another doctor. So when I had some, discussion of homeopathy, I said it's a high time I give him lecture. So I was really impressed that today you have taken my heart.
Thank you.